Going. Thank you very much, Claudia, for agreeing to give a Build a Cell seminar. And you can get started.
For inviting. Yes. Okay. So I'll first share the screen so that I don't try to do two things at the same time. Okay. Hi everyone. My name is Claudia Bonfio, and I was asked to introduce myself, and so I'll spend a few slides driving you through, walking you through my path, if I can actually manage the slides to move. Okay, so this is a map of Europe, and I'm from Italy. I'm from Tuscany, which is in the center of Italy, and I moved to do my PhD at the University of Trento, which is a relatively small city in the north, close to the Austrian borders, close to the Alps. And that's where I did my PhD, working on the synthesis and the catalytic activity of iron sulfur clusters. And so the PhD topic was already addressing the major question, the main question that has basically accompanied me throughout my whole scientific career, which is how life started on our planet. And so when I was a PhD student, I was in Sheriff Menzies group, and we started looking into, let's say, the origin of proteins and metalloproteins. And I was very lucky because this project on iron sulfur clusters gave me the chance to spend some time also abroad. And so I spent 4 months in 2015 at the MRC Laboratory of Molecular Biology in Cambridge, UK. And then I spent 3 months in 2016 in the other Cambridge at Harvard, where I actually overlapped with Kate moving for I think 1 week or something. And so this was my PhD. And at the end of my PhD, I decided that I definitely wanted to keep working on origin of life research, but I wanted to do something slightly different. So I decided to move to the UK. Moved to Cambridge, where, as I said before, I had already spent a few months doing my PhD. But here, instead of looking into peptides, metallopeptides, and proteins, I looked at one of the other building blocks of life, which was lipids. And here I looked at the synthesis, the prebiotic synthesis of lipids. I looked at how lipids assemble into lipid membranes. And then I also tried to learn a bit how these lipid membranes can become functional, moving from the LMB to the University of Cambridge in Chris Hunter's group. And then basically at the same time when the pandemic hit, I also decided that I wanted to become independent. So it was the worst time ever to search for a job, but I was very lucky. And so, during my postdoc, I also spent some time in Harvard. So that was still to work on primitive cells. And so, at the end of my postdoc, I was very lucky. And I applied for a position at the Institute for Supramolecular Science and Engineering in Strasbourg. And this is an institute that was funded about 20 years ago by Jean-Marie Lehn, Nobel laureate for supramolecular chemistry. And I felt like this was a great place where I could learn a lot about supramolecular chemistry, and at the same time I could apply what I was, what my colleagues were doing, and from the interactions and so on, I could apply this to origin of life research and trying to understand how primitive cells emerged and evolved. And so basically what we try to do when we try to understand how life started on our planet is we try to think how we can recreate life. And the problem is that something that Albert Eschenmoser said a while ago is that it's difficult to think of ways to understand how life started in terms of discovering how life started, because we don't have a way yet to go back in time to about 4 billion years ago when life actually started. And so what we can try to reinvent the origin of life and life in the lab. And this is what probably connects all of us here, and in terms of the interest that we have, but also all the different seminars that this amazing series that I'm very happy to be contributing to is talking about, which is how living cells work, what we can learn from them, how we can engineer them, and how we can build them from scratch. And so basically, I'm a chemist by background, but as you might have realized, I'm very interested in how biology works. So the way we have in the lab to understand how life might have started is trying to connect chemistry and biology, trying to go from something that molecules, chemical reactions that we definitely would not consider life to something that we definitely would consider life. And so we have different ways of doing that. And so I thought that something that was needed was a bit of a definition. And I decided to share with you the definition of the term protocell. And the reason is that we talk about synthetic cells, we talk about artificial cells. What I talk about the most, and what I'm going to talk about a lot during this talk, is what a protocell is. And so I thought that maybe sharing this with you was quite relevant. And so a protocell, according to what Caparis Mirazzo said and wrote in the Encyclopedia of Astrobiology about a decade ago, is that a protocell is a model that involves a compartment linked to chemical processes. So there's a compartment with chemical processes that are taking place around the compartment and inside the compartment. And this model, that could be both experimental or theoretical, aims at explaining how life started and how modern cells formed on early Earth. So if we think about how a primitive cell would have looked like, this is what we tend to think. We tend to think of as a very simple compartment with very simple chemical reactions, potentially reaction cycles or networks taking place inside of that. And this is the first big question, because we don't know how this started. And then the second question is, if we look at how modern cells look like, they are way more complicated. If we look at just the membrane is made of a large number of different amphiphiles, and it's not just amphiphiles, it's not just lipids. There are a variety of other molecules and proteins composing the modern membrane. And so when we look at these two systems, this primitive cell and modern cell, the first big question is how a primitive cell formed, and the second question is how a primitive cell became a modern cell. So again, if we start looking at just what a primitive cell would look like, we said that it would be something very, very simple. What do we mean by simple? So if we look at just the lipid membrane that forms this that forms this primitive cell, we can see that the way I've drawn that is made of one single drawing of an amphiphile that gets repeated. And the reason is that most of the examples we have of studies of primitive cells usually involve a relatively small number of lipids, usually fatty acids. And so if we look at what modern cells have, modern membranes will have much more complex lipids, usually most of them are phospholipids. So the first question that we tried to answer when I was still in Cambridge is, how did early lipids evolve into modern lipids? And so we can see that this as a sort of treasure hunt map where we have phospholipids. This is just a representation of a phospholipid, a modern phospholipid that we have. And you can see that it can be sort of divided in 4 different parts. If we go from left to right, we have acyl chains that I have here abbreviated with R1 and R2. We have acyl chains, then we have a light blue part, which is glycerol. Then we have phosphate, and most of the times phosphate is further derivatized with additional small molecules. In this case, I've drawn ethanolamine. So this is the kind of structure that modern phospholipids have. And this is the structure of a very simple prebiotic lipid, which is a fatty acid. So how do we go from one to the other? especially considering that we need to combine together 4 different building blocks. And so there's already some studies that have been published trying to combine some of these building blocks together. For example, glycerol and phosphate. There's a paper from a few years back from the Sutherland group that shows that you can relatively easy phosphorylate glycerol, and you end up with glycerol cyclic phosphate that can be opened into glycerol 1 and glycerol 2 phosphate. So let's say that we have combined these 2 pieces together. How do we start combining the third one? This is something that I've done at the beginning of my postdoc in Jan Sutherland's group. And we simply took a glycerol phosphate moiety and we tried to acylate that. So we tried to add some acyl groups to it. And what we ended up with was a mixture of mono and bis acylated species. So just to give you an idea, when we look at modern phospholipids are mostly bis acylated, so they will have 2 lipid chains. So we were getting a mixture of mono and bis acylated glycerol phosphates. The main problem, though, is that when you have short chain acylated glycerol phosphates, they form very efficiently, while if you want the long chain ones, they don't form that efficiently. So we needed to find a way to accumulate the long chain amphiphiles over the short chain species that do not accumulate, that do not self-assemble. And so we came up with this idea of a cycle in which we would start from fatty acids, which we said are the building block, the starting material that we have to make for modern phospholipids. We would activate them, making these acyl imidazolides that you can see on the top of our cycle. We would use those to acylate glycerol phosphate. And then the key point was that when we were the short chain acylated glycerol phosphates that we were making would get relatively easily hydrolyzed, and the long chain ones would start accumulating and would self-assemble into lipid membranes. So this was a sort of way that we found to accumulate selectively, length selectively, amphiphiles on early Earth through recycling. So I thought that this was not only a good chance for me to show you what I've done in these past years, but also to possibly discuss about problems that still need to be fixed. And you'll see that throughout the presentation, I'll highlight a few problems that I think we still need to fix, especially when we talk about lipids and lipid membranes on early Earth. And so the first problem that I want to mention is that those of you that have that are familiar with glycerol phosphate might have noticed that here I have drawn glycerol 2 phosphate. Glycerol 2 phosphate, I hope you can see my arrow, has the phosphate group on the hydroxyl, on the central hydroxyl of a glycerol moiety, the hydroxyl in position 2. Now, the reason we use this substrate is because it's very reactive. The problem with this substrate is that it is not a biological relevant substrate, because all lipids that we have in modern phospholipids are either glycerol 1 or glycerol 3 phosphates. And so the main problem that I thought I would highlight here is that we need to understand how we can select for the correct head group over the wrong one, even though it's more reactive. So if we go back to our treasure hunt map, let's say that we have found a way to make, to combine 3 different building blocks, even though I've already told you that it wouldn't be exactly the correct isomer. Now, the last point that still needs to be done, even though there are some studies that have shown how you can add the last small molecule to derivatize phosphate, there's still a lot that needs to be done in this direction. And so even also here, I just wanted to highlight some problems that hopefully will be fixed in the coming years, and hopefully some of them from my lab. One problem is that the conditions that are currently used for acylation And so to add the lipid chain are quite often involved in organic solvents. And we know that in terms of trying to build life from scratch in an origin of life context, it's quite difficult to think of organic solvents, especially high amounts of organic solvents, to be used for chemical reactions. Second problem is that unless we, if we want to find chemical processes that can form fatty acids in a controlled way, so if for a moment we exclude the Fischer-Tropsch synthesis of long chain alkanes, alcohols, fatty acids, and so on, we still are far from being able to control not only the length of the fatty acid chains that we are building, but also the positioning of double bonds. And we know that modern phospholipids usually contain at least one double bond in their chain in a very specific position. Third problem is that, as I was saying, there are some studies that show that in dry conditions, using wet-dry cycles, you can achieve the formation of these phospholipids. So you can add this small molecule to the phosphate moiety, but there's not enough, and there are no reports that show how you can build all the different head groups of modern phospholipids that I have mentioned here. So PC would say for phosphatidylcholines, P for phosphatidylethanolaamines, phosphatidylserines, phosphinosytols, and phosphoglycerols. Another point, everyone basically, including ourselves, we are focusing on ester-based phospholipids, which are the most common lipids present in bacteria and eukaryotes. But archaea use completely different types of ethers. One of the main differences being that they are ether-linked and not the fatty acids, the alkyl chain is ether-linked and not ester-linked to the glycerol phosphate moiety. So this is something that would require a lot of study as well. And then there's, I've written about, I've included polycyclic hydrocarbons here because those of you who have looked at the composition of modern membranes would know that usually modern membranes are not made of just phospholipids and they are not made of just proteins, but usually there are some other molecules, so for example, cholesterol and sterols in general, or in bacteria, hopanes and derivatives that contribute to the stability, lipid packing, morphology of the lipid membrane. And this is something that potentially derives from polycyclic hydrocarbons, maybe not, but that could be very interesting to understand and explore a bit further. So the first part of this talk was about what we've done and what we still need to do in terms of synthesizing lipids that could have formed lipid membranes on early Earth. And so the next step for me was to understand, once we have formed these lipid membranes, are they just inert? Would they just, be spectators to any chemistry that runs around them, or would they participate to that? And so what we decided to look at was some chemistry that was being developed in John Sutherland's group while I was there, which is what we call activation chemistry, which basically exploits the ability of methyl isocyanide to simultaneously activate carboxylates and phosphates in solution. And this would yield not only polypeptides, potentially RNA polymerization, but also mixed species in which you activate the amino acids, the carboxylates, for example, those present in amino acids and peptides, and form amino-acylated or peptidylated DNA or RNA species. So this was a chemistry that had been developed in the lab. And as I mentioned, it works on carboxylates, and the carboxylates that at the beginning we were interested in were those present in amino acids and peptides. But I've just told you that one of the key ingredients of lipid membranes on early Earth was likely fatty acids, which have a carboxylate as well. And so what we wanted to know was whether carboxylic acids present in fatty acids would interfere with this chemistry or not. So we started from something very simple, and we took a nucleotide and we just mix it with a solution containing solution containing lipid membranes made of fatty acids, and we expose them to this activation chemistry. And what you can see here are NMR spectra. On the top, you can see the NMR that results from the reaction run in the absence of lipid membranes, so just with nucleotides and methyl isocyanide, so this activation chemistry. What you can see is that we have almost entirely, basically entirely conversion of the nucleotide to its cyclic form, which is the activated version. And what you can see on the bottom is that we have two extra peaks when we have lipid membranes there. One peak, which is the central one, it's just a bit of starting material, so the reaction is not fully completed yet. But the other peak that you can see is possibly more interesting because it derives from the activation of the carboxylic acid present on the membrane, so fatty acids in this case, and the formation of a derivative, which is an acylated nucleotide. Now, what was interesting about this molecule is that we have basically combined something that has an alkyl chain with something that is polar. And so this resulting derivative, this resulting conjugate is an amphiphile. And so we decided to explore what this amphiphile would do. The first thing that we looked at was whether this amphiphile would insert into lipid membrane and drive their growth. And this is exactly what we found. So on the left, you can see the control experiments when you add some micelles made of fatty acids to membranes made of fatty acids, and you can see that the surface area increase. And what you can see on the right, the blue bars show that when you add these lipid-nucleotide conjugate to lipid membranes made of fatty acids, they grow as well. And this is not just something that happens when you make lipid-nucleotide conjugates. The same can be achieved when you build in the same way lipid-amino acid conjugates. Now, what's interesting about amino acids, though, is that they have side chains, and depending on the side chain and the amino acid that you pick, the side chain will have different functional groups. So in this case, we decided to use arginine because it would bear a positive charge. And if you have a lipid conjugate that bears a positive charge, then basically you are localizing positive charges on the lipid membrane. And if you are localizing positive charges on the lipid membrane, then you can potentially have those positive charges interacting with something that is negatively charged in solution. And so what we were curious about was whether our lipid amino acid conjugates could localize to the membrane and also drive the co-localization of RNA to the lipid membrane. And this is what we could see by microscopy. So if you look at the bottom images, you can see that we had co-localization of fluorescently labeled RNA to the surface of the lipid membrane, meaning that these lipid-based conjugates not only can drive the growth of primitive cells, but they can also mediate the co-localization of different biomolecules. Now, again, as I've done before, we still have some problems to fix also in this context. Also, when we move from the synthesis of the lipid membranes to combining these lipid membranes with chemistry, prebiotic chemistry. So the first problem that still needs to be fixed is what happens if you have not just one chemistry that you want to test with membranes, but what if you have multiple chemistries that you want to test? How much do we know about the orthogonality of all the different chemical reactions that have been explored in prebiotic chemistry? And the second point is related to the chemical compatibility between the lipid membranes and these reactions, because we've seen here an example in which the components of the lipid membranes take part into the chemical reaction and participate and yield potentially useful species. But how can we control this reactivity of the molecules forming the membranes is something that we still need to investigate. So I've told you that I was interested in looking at how membranes behave in the presence of different chemistries, but also on a simpler scale. I was also interested in understanding how lipid membranes behave when you simply change the conditions, the environment. And so what happens if, for example, you change, you expose these membranes to different gradients? Now, the gradient the conditions I decided to look at were temperature. And the reason was that there was a paper that my PhD supervisor, Sherif, published when he was in Jack Shostak's group during his postdoc. And this paper was about how fatty acid-based membranes would react at different temperatures and whether they would release all their content or not at some temperature. So what you can see here is a graph that I took from that paper, and you can see that they tested a range of different fatty acid-based membranes. They had encapsulated nucleotides inside these fatty acid-based membranes, and what you can see is that depending on the fatty acid system that they studied, at some temperature they would have a complete release of the nucleotide that was originally contained inside the membrane. And they not only showed that this phenomenon would depend on the type of fatty acid that was used. For example, shorter chains, DA stands for decanoic acid. Shorter chain fatty acids would release the content more efficiently than oleic acid-based membranes. But this was also depending on the size of the molecule that was contained inside the lipid membrane. So here you can see that a dinucleotide would be released more easily than a longer polymer. So I was very curious about this, but I was much more curious about what was happening to the membrane when increasing the temperature and when changing the temperature of the solution. And so what I decided to do was to look at what happens under the microscope. So this is the micrograph of what you start with, lipid membranes made of fatty acids. In this case, we use myristoleic acid as a model, but exactly the same situation happens for decanoic acid-based membranes. And so what hopefully you'll be able to see now is a movie that for me just got stopped. Okay, where we have fluorescently labeled lipid membranes, and you can see that we are increasing the temperature, and you'll see that at some point at very high temperature, the morphology of the systems that you were looking at, of these fluorescent systems that you were looking at, changes completely. Now they are, when we keep them at 95 degrees, they keep fusing together, they keep merging. They look very different compared to what they were at the beginning. And now you can see that we are decreasing the temperature. And at some point they become very unstable. And you can see that from their surface, they start to bud out new lipid membranes. So these are just some pictures that I took from the same video, just in case the video would work. So you can see on the left, it's our starting point. We have lipid membranes that are fluorescently labeled. And then we go we increase the temperature. And if you look at the pictures on the right, we have these very large, dense structures, fluorescently labeled structures that look like oil droplets. And when we cool down the system, what you can see is that, as I was saying, from the surface of these oil droplets, you can observe budding of new lipid membranes that get released in solution. So we decided to look at this also from with other techniques. And the first technique that we used was absorbance, because we know that lipid membranes and oil droplets would scatter the light in a very different way. So you can see, if you look at the gray line here, at the gray profile, you can see that the absorbance is quite stable until we get to around 60 degrees. And at that point, we have a very strong increase in absorbance. The same results you can get from using a dye. In this case, we use Lordan, which is sensitive to lipid packing. And so what you can see is that the purple line, the purple profile here, shows a very similar situation in which you have a more or less stable readout of this dye, of this fluorescent dye. And then at some point you have just a drop in the, a change in the fluorescence emission of this dye. We also decided to look at what modeling could tell us about this. And modeling proved that we were not just increasing the lamellarity of the lipid membranes, because the increase in absorbance was just way too much. But what we were actually doing, the only way to explain that such an increase in absorbance was if we had very giant lipid-dense structures, oil droplets. So this is the situation that we have. At 25 degrees, we have lipid membranes. At 95 degrees, we have oil droplets. Why is that? Well, the reason we found out was relatively simple. For those of you who have not worked with fatty acids before, you would know that in order to form lipid membranes with fatty acids, you need to have half of your fatty acid molecules as deprotonated and half of them as protonated. When you have all the fatty acid molecules deprotonated, the lipids form micelles. If all of them are protonated, they would form oil droplets. So basically here, we were going from having a situation in which we have a mixture of protonated and deprotonated fatty acids to a situation in which we had only protonated fatty acids. And so if we look at the at this graph that I found in literature for oleic acid, even though it's not, and it works very similarly for myristoylic acid, is that basically the only way we had to explain this change in phase of our fatty acid moieties, of our fatty acid structures, was if we were going from having a mixture of myristolate and myristoylic acid, so deprotonated and protonated form, to a majority of protonated version, which would only happen when the pH in the solution decreases. And that's when we found out that there are some buffers that are not stable with temperature and whose pH decreases with temperature. And we found out that a relatively large number of even prebiotically plausible buffers change pH when you increase the temperature. And so basically what we were observing was a change in pH that was concomitant with the increase of temperature. And so a disruption, a phase transition from fatty acid membranes to fatty acid oil droplets. And so a collapsing of the membranes into oil droplets that would be reversible, and so lipid membranes could form upon cooling. Now, we were quite interested in whether we could modulate, control this phase transition and the temperature at which this phase transition would occur. And so we decided to explore the role of the effect of salts. And what you can see here is that when we were adding some salt, we decided to test potassium chloride. The temperature at which this phase transition, this collapsing of lipid membranes to oil droplets occurs is lower compared to when we do this experiment in the absence of salt. We decided to look at nucleotides, same effect. The moment you add some nucleotides to the solution, this collapsing of the lipid membranes occurs sooner. Same goes for peptides, and we tested the peptides that were not bearing any charge and peptides that were bearing any charge. Now, this was the result that we had. We were able to reversibly convert lipid membranes into oil droplets. So what we decided to do was try to find an advantage for this. And so we decided to look back at what happens to the content of these liposomes that we are forming. And so what you can see on the left is the starting situation. We had encapsulated some fluorescent dye inside the lipid membranes. It gets completely released when we increase the temperature to 95 degrees. But what you can see is that when we cool down the system, some of the liposomes that get reformed are re-encapsulating the dye. Now, just something that I will mention now, because I always get this question, so I'm just preventing this to happen, but I'm happy to discuss in case it's needed, is that you can see in the picture of on the right that the background is very low, so it's very low, has very low fluorescence compared to the fluorescence inside the lipid membrane. And this is just, let's say, an artifact due to the fact that we were diluting the solution in order to see if some membranes, some liposomes were fluorescent. So if we had not diluted the solution, we would probably not be able to distinguish that well between lipid membrane, what happened between the content of lipid membranes and the outside. Now, what we decided to do was then to start mixing different populations of liposomes that had different dyes to see if we could mix the content of these different populations. So we took two populations of membranes that had two different dyes, mixed them, exposed them to this thermal cycle, and the outcome we got was a new population of lipid membranes that would contain mixed content compared to their parent liposomes. Same goes for the membrane composition. So if we start from lipid membranes with different dyes and we mix them with different lipids, we mix them, we expose them to the thermal cycle, the new population, the daughter liposomes now have mixed composition compared to the parent liposomes. So we decided to see if we could do also something a bit more useful with that. So we decided to reconstitute an RNA aptamer. So the idea here was to start from two different populations of lipid membranes that would contain each one part of an aptamer. And then we would mix them and expose them to thermal cycle. And if we, upon addition of DFHBI, that would be bound by the RNA aptamer and emit fluorescence, only if we had reconstituted the two half RNA aptamer, the two halves of the RNA aptamer inside the same liposome, we would be able to observe some fluorescence. And this is what we could observe in our measurements. And so the main point here, the main message here is that we were able to use this reversible degradation and reformation of lipid membranes in order to mix the content of the parent liposomes and reshuffle this content and forming thus daughter liposomes, daughter primitive cells that had now mixed content. Now, again, I will highlight an issue with this, something that needs to be fixed. So let's say that this was something that could have happened, even though we can, to some extent, modulate and control this, it's still something that would not be advisable in the long term, because what we would like to have is what happens, is what we have right now in modern cells, so stable lipid membranes, and then a machinery that sits on the membrane and that can control the transport of molecules rather than destroying the liposomes, destroying the lipid membrane, and then just try to re-encapsulate everything. So another problem that needs to be fixed is a way to achieve a controlled transport in functional primitive cells. And so what I think we need to look into in the future is we have now understood that we have a lot of there's a lot of research that has demonstrated different ways on how the different building blocks of life could have emerged on early Earth. we have different prebiotic synthesis for nucleotides, we have different prebiotic synthesis for amino acids, we have different prebiotic synthesis for lipids. We also have some prebiotic processes that allow oligomerization of amino acids and nucleotides and self-assembly of lipids. So what comes next? Can we combine all of these things together in order to build more functional, more advanced primitive cells that are capable of the same activities that modern cells are capable of? For example, transport, signaling, catalysis on the membrane, and interaction with other primitive cells. And with this, I will, let's say, open the floor for questions, but I will first thank everyone who has contributed to the research that I've presented today, which is mostly people from the lab in Cambridge, but these are also the amazing people I had the chance to work with in this last year in Strasbourg, and so hopefully soon I'll be able to share with you also some of the new things that we are doing all together. Thank you.
Thank you so much. That was a really cool talk, especially for me being a liposome person. I really love it. There are some questions in chat. Can you see them?
Yes.
If you could read the question before answering it, that's going to go for the recording.
Sure. Okay, so first question says, could this lipid synthesis you describe be catalyzed by some really primitive enzymes, not done entirely non-enzymatically, but with some earliest possible catalytic RNA or peptide? So the answer is that I don't know. It would be really cool to start combining things together also in that sense. So the main approach in the origin of life research has been in the past to basically only focus on one specific individual building block. And I think now people are starting to realize how we need to combine things together. So it would be really cool if some enzyme, if some peptides are found to be able to catalyze acylation reactions or phospholipid head group synthesis. As far as I know, there's nothing out there yet on this, but it would be really cool if that could be possible. I'll move to the next question. So the next question is, would this temperature-dependent fusion possibly be a prebiotic growth and division mechanism? You could imagine this happening around hydrothermal vents. So the fusion part, so if this refers to the fact that at high temperature we were observing fusion, this was something that was happening on oil droplets. So after we have the fatty acid membranes have transitioned to oil droplets and have collapsed into oil droplets. So in that sense, yes, it would be considered growth, but it's not the growth that we would associate with liposome growth, which then potentially would drive to division. Having said that, what we observed was that this, in general, this reversible phase transition between lipid membranes and oil droplets is something that can occur very easily, assuming you are in a buffer that is sensitive to temperature, and so definitely it could occur in a lot of different environments, quite likely on early Earth, including hydrothermal vents. Okay, I'll read the next question. They say excellent talk. Thank you very much. Does the membrane dynamics with thermal cycling is concern for fatty acids, phospholipids, and membrane with cholesterol, or is it unique to some specific type of lipids? Yes, it is definitely only specific for fatty acids, to the point that when you go for longer chain fatty acids, for example, oleic acid, it doesn't happen anymore unless you go to very high temperature. So it's definitely very lipid dependent. It does not happen for phospholipids, and even just pure phospholipids, even in the absence of cholesterol, even more so with cholesterol. So It is definitely something that would have happened for decanoic acid-based vesicles. Some of our results show that with decanoic acid vesicles, you would have these collapsing into oil droplets, even at room temperature, in the presence of some peptides. So definitely it would have happened for simple fatty acids and short chain fatty acids. The more you go for something complex, the less it happens. Next question. Well, quite a lot of questions. Okay, I was worried it was way too chemistry. Anyway, okay, so they say, thanks for the great talk. Thank you very much. Would you speculate what was the lipid composition for Luca? Was it phospholipids or something simpler? So I don't have an answer for this. What I would say is that I believe it was probably something more complex than fatty acids, because we all know that there are a lot of problems with fatty acids, that we can find ways to avoid things, but in general, we need to find a solution for fatty acids. So I'm a strong believer in phospholipids. So if I had to bet my money on that, I would say that phospholipids were key at some point before the emergence of living cells. But as far as I know, there isn't a clear consensus on the lipid composition in, let's say, primitive cells or at the Luca step. Next question. Could there be some simple glycosidation added to those simplest membranes or any other lipid derivatives, or do you think it should be pure lipid membranes? I absolutely do not think it should be only lipids. Something that I'm very intrigued by is that it's probably something that I did not stress upon too much at the beginning, but it's fascinating how lipid membranes are modern membranes are basically composed of 50% proteins and 50% lipids. And every time we make these model primitive cells, we use 100% lipids. To me, it doesn't make any sense. And so absolutely not to the pure lipid membranes. It would, so some of the work I've shown shows that lipid fatty acids, for example, take part into the chemistry that we run around them and forms some derivatives. So it would be very interesting to see what kind of derivatives we can build with the different types of chemistries that have been developed in prebiotic chemistry. So yeah, it could be anything. It would be very nice to explore that. Okay, last question so far. Great work, thanks. Could presence of mineral surfaces help or inhibit the transition between lipids and oil droplets as you do thermal cycling? It's a good question. I don't know. Definitely, there's quite a lot of research that shows that mineral surfaces can help in the self-assembly of lipid membranes. So maybe I would tend to think that they could participate in a positive, do something positive rather than inhibit. But in general, but it is possible that mineral surfaces would play a role in this. I haven't explored that, so it would be very interesting to try both in the presence of the thermal cycling and without, to see if there's an effect of mineral surfaces in the transition between membranes and oil droplets. It would be very interesting to try. Yeah, that's it.
I just want to say thank you so much. And I was very intrigued by the statements you made about modern membranes not being pure lipid membranes. I always find that fascinating too, that we don't do that work. I mean, I was trained doing pure liposome work too. And I think this is something where we need to go, is look at short peptides, RNA, like the stuff you were doing about RNA association with membranes. That's really fascinating.
Yeah, no, yeah, I think it's, I think I think there's just a lot that we could explore. What I really don't like is when people assume that lipid membranes would just be, inert compartments, and you can just develop any kind of prebiotic chemistry process that you want, and then you just need to put it inside liposomes, assuming that it's very easy to do so, and that's when problems start. So I think lipids should play a much bigger role in this, and the molecules. There's a lot of chemical reactions that it's easier to run chemical reactions in water, right? And so this doesn't really help in building molecules that would localize to the membrane or that could do something to the membrane. But I think there's just a lot of chemistry that would be very interesting to explore in a prebiotic context, that would happen at the interface between water and membrane, so on the membrane surface. So yeah, well, I have a whole career to build, so there's a lot to explore there.
Yeah, definitely, we should be looking at more of that. All right, so if there's no other questions, I want to say thank you so much, Claudia, again.
I'll just stop so that I can see you a bit bigger. Yeah.
And thanks everyone else, and see you all next week.
Thank you very much again for the invitation.
Bye.